Welcome back to study section 6 on seed dormancy and germination, development of mineral nutrition, growth regulation, a continuation of what you've learned in the previous study section. In the previous study section, you've learned that crop physiology is the study of the plant processes responsible for the growth, development, and production of economic yield by crop plants. In this study section, you will be introduced to other aspects of crop physiology, which includes seed dormancy and germination, development of mineral nutrition, and growth regulation. At the end of this study section, you should be able to describe seed dormancy and germination. 2. Discuss the development of mineral nutrition. 3. Discuss growth regulations in plants. Now, let's discuss each of the learning outcomes one after the other. Dormancy is a condition in viable seed which prevents it from germinating when supplied with the factors normally considered adequate for germination. What are the causes of seed dormancy? Causes of seed dormancy are 1. Impermeable seed coats. This is an example of structural constraints in germinations of seeds. The seed coat may be impermeable to water or gases, thus limiting metabolic activity in the seed. Dormancy of this type is broken when the tester becomes less resistant. Such methods include mechanical or chemical scarification. 2. Rudimentary embryo. This results when non-germination is caused by shedding of seed before they are morphologically mature. 3. Chemical inhibition. The mechanism of inhibition is by blocking the activity of the enzyme necessary for starch and protein breakdown. Chemical inhibitor can be removed by soaking the seeds in water or other chemicals. Advantages of seed dormancy includes 1. It prevents germination of seed until when planted. 2. It also prolongs viability of seed in the storage. The disadvantages are 1. It prevents prompt germination and uniform emergence of seedlings. 2. It interferes with planting schedule. Seed germination is the process whereby dormant embryo wake up from their sleep. It is the awakening of sleeping dormant embryo. Process of seed germination includes 1. Water imbibition. Water is absorbed through natural opening in the seed coat. The water causes the cell to become turgid and the seed expands in volume, thereby making the seed coat more permeable to oxygen and carbon dioxide gases. As swelling occurs, the seed coat ruptures and this facilitates both water and gaseous exchange and the emergence of the seedling. 2. Enzyme activation. Enzymes are activated in the presence of water and are mobilized and are used in breaking down stored food materials. The stored products are converted to more soluble materials. 3. Initiation of embryo growth. The food reserve is used to produce new material, primarily for the shoots and roots. 4. Rupture of seed coat and emergence of seedling. Rupture of the seed coat is caused by swelling or sometimes emergence of plumule and or radicle. 5. Establishment of seedling. Seedling is established when it starts to absorb water through the primary roots and is able to manufacture food through photosynthesis. Another learning outcome is the development of mineral nutrition. Nutrition is the process of synthesizing food, its breakdown and utilization for various functions of the body. The chemical substances in food are called nutrients. Example, water, minerals, carbohydrates, protein, fats. Mineral nutrition is the study of how plants get mineral elements and utilize them for their growth. Apart from carbohydrates, which includes elements such as carbon, hydrogen, and oxygen that are synthesized by the process of photosynthesis, a variety of elements are needed for survival. Criteria used in determining essential elements are 1. An essential element is absolutely necessary for normal growth and reproduction of the plant and should be a part of essential metabolites for plant growth. 2. The requirement of the element is very specific and it cannot be replaced by another element. 3. The element is directly or indirectly involved in the metabolism of a plant. Now we'll move on to types of essential elements. We have 1. Microelements, also known as macronutrients, are elements required in minute or small quantity, and they are also called trace elements. Examples of micronutrients are 1. We have boron, cobalt, 3. Copper, 4. Iron, then 5. Zinc. Another type of essential element is macroelements, also known as macronutrients. They are elements required in relatively large quantities. Examples are carbon, hydrogen, oxygen, phosphorus, and potassium. Then we'll move on to growth regulation in plants. Plant growth regulators usually are defined as organic compounds other than nutrients that in small concentrations affect the physiological processes of plants. In practical purpose, they are defined as either natural or synthetic compounds that are applied directly to plants to alter its life processes or structure in some beneficial way so as to enhance the yield, improve quality, and facilitate harvesting. They are classified as auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Auxin, which is the first plant hormone or growth substance discovered. Compounds are considered auxins if they are synthesized by plants. Functions of auxins are 1. It stimulates cell loosening, expansion, and elongation. It initiates adventitious roots on stem cuttings, and 3. It stimulates differentiation of phloem and xylem. Another growth regulator is gibberellins. Gibberellins are synthesized in young tissues of the shoots, uncertainly in roots and also in developing seeds. Functions of gibberellins are 1. It stimulates cell elongation in stem. 2. It breaks seed dormancy in some plants, which requires light for generations. And 3. It plays a role in development of seedless fruit. Cytokinins. Cytokinins, these are the compounds resembling with adenine which stimulate cell division. Their functions are 1. It stimulates cell division. 2. It stimulates morphogenesis in tissue culture. 3. It stimulates the growth of lateral buds and leaf expansion by cell division and enlargement. Then we'll move on to abscisic acid. Abscisic acid controls several physiological processes in plants. It also controls seed germination, vegetative growth, and bud dormancy. Their functions are 1. It stimulates the closure of stomata. It induces seed and bud dormancy. And 3. It induces seed to synthesize storage protein. Thanks for listening. You can read more about the study section in the study material.